there. Okay. I'm Mrs. Brodigo. I teach at Cahokia High School. I want to invite you into our cell. This is my AP Biology class, and we have created a cell for you. Each student is focusing on a structure or an organelle, and they're going to describe the structure, the function, and the relationship of those organelles. Hope you enjoy it.
Enjoy!
The structure of the cell dying presenting is cilia. Cilia are like hair-like little appendage that stick out from cell. They whip back and forth and help move around in cellular fluids. The function of the cilia is moving. They can be found in the respiratory tract, nose, and middle ear. Cilia will wave in a rhythmic pulsing motion to keep internal passageway free of mucus or foreign particles. The only relation that cilia has is flagellum, since both are located outside the cell and both involve movement.
My organelle is the flagellum. The flagellum is composed of membrane-enclosed microtubules. It has a core of microtubules and an extension of the plasma membrane. It contains 9 doublets of microtubules. Each doublet joins another microtubule to form a ring of 9 triplets. The flagella has a repetitive motion that generates force that moves in the same direction the flagellum's axis. By the way, this is a composite cell. You would never find the cilia and the flagella in the same cell.
The cell membrane is a phospholipid bilayer. The phospholipid bilayer is made of hydrophilic phosphate heads and hydrophobic lipid tails. Water surrounds the phosphate heads, so water is on the inside and outside of the cell membrane. Cholesterol molecules are embedded in the cell's membrane to help make the membrane less permeable. We use small red cups to help represent the cholesterol. The cholesterol goes through the cell membrane, which stabilizes it.
This door represents the channel protein. It is located on the cell membrane. This protein is responsible for transporting ions. It allows some things to come in and excludes others.
My organelle is called the mitochondria. Mitochondria are found in most eukaryotic cells. The mitochondria is bean-shaped and it is consisted of a double membrane, which is known as the phospholipid bilayer. The outer layer is like a skin for protecting the cell, and the inside is made up of cristae, or crista for singular. The mitochondria consists of maternal DNA, which are these little structures, and glucose and oxygen enter through the mitochondria from its outer membrane, or the phospholipid bilayer, and it goes through a phase known as cellular respiration. During cellular respiration, ATP energy is formed, adenosine triphosphate is created, and it stays within the cell, but it leaves the mitochondria and helps the cell function.
The endoplasmic reticulum, ER for short, is a network of membranes throughout the cell and connected to the nucleus. There are 2 types of the ER, both rough and smooth, have the same types of membranes, but different shapes. The rough ER has ribosomes, and the smooth ER has layers. The ER also provides the protein quality control function. Proteins are usually retained in this compartment until they require the conformation.
My structure is the nucleus, which is located in the heart of the cell inside the cell's cytoplasm. The nucleus consists of chromatin, pores, which are these structures on the outside of the nucleus, which allow RNA and protein to pass through the cell. The nucleus also includes nuclear envelopes, which keeps all chromatin and the nucleolus inside the nucleus. And the function of the cell is to store the cell's DNA and coordinate cell activity.
My organelle is the ribosomes. They're located in the cytoplasm and on the rough ER. The ribosomes synthesize proteins within the cell. They are made up of small pieces of RNA and protein. A perfect example of a ribosome are enzymes that are found in the lysosomes. I represented the ribosomes with yellow and brown cotton balls. The yellow cotton balls represent the free-moving ribosomes, and the brown cotton balls represent the ribosomes on the rough ER.
Small organelle is the Golgi Apparatus. It has a unique maze-like structure made of flattened membranous sacs called cisternae. The Golgi's job is to modify, sort, and package proteins, which are these little purple balls in here. It relates to the other cell because the endoplasmic reticulum sends proteins that it has created into the Golgi to be perfected, and then the perfected proteins are then released into the vesicles, which are these things. The vesicles then transport the perfected proteins along the microtubules to the channel protein, so that way it could be sent out of the cell and into the body.
My organelle is the lysosome. The lysosome structure consists of membranous sacs of hydrolytic enzymes, which are these. The enzymes break down molecules such as mitochondrial particles. The mitochondrial particles are then recycled and used to create more mitochondria, which makes ATP energy that is imperative for cell life. I use the recycle symbol because the lysosome is the structure in the cell that is responsible for the remaking and reusing of other cell organelle particles.
In our eukaryotic composite cell, peroxisomes are small membrane enclosed organelles that contain enzymes involved in a variety of metabolic reactions, including several aspects of energy metabolism. Peroxisomes have single membranes of phospholipids. They break down toxins such as alcohol. H2O2, known as hydrogen peroxide, breaks down into H2O, known as water molecules.
In our eukaryotic composite cell, the cytoskeleton, which includes the microfilaments and microtubulars, are made up of small units that self-assemble to create movement and structure all throughout a cell. The vesicles, which are along the microfilaments, connect to the Golgi apparatus. This brings the vesicles to the channel protein. I represented the microtubulars with the bright pink string and the bright pink streamers, and the microfilaments with the white ribbon.
This is the organelle that I made, which is a centrosome. The centrosome is made of 2 centrioles, which is a 9+3 arrangement of microtubules in a circular manner. They provide structure for a cell and line the chromosomes up and pull them apart, forming daughter cells because the chromosomes cannot move by themselves. They are found in animal cells near the nucleus. The pores of the nucleus let chromosomes exit and pull them to the opposite side to form new cells. The last organelle we're going to talk about is the cytoplasm. We couldn't really find anything to use to symbolize the cytoplasm because it would take so much space up in the room, knowing that it is about 60 to 70% water. The cytoplasm is the region between the nucleus and the plasma membrane of the cell, and the cytoplasm is the substance in which all of the organelles are located.
Okay, we hope that you enjoyed your tour of our cell, and even more importantly, we hope that you learned something. We certainly did, and we know it's not perfect. We're not saying that it is, but we're going to put the students on the spot here and ask them what they thought about this learning experience.
Well, honestly, I feel that it was very effective because first I really didn't understand about the different organelles in the cell, but after the weeks and weeks and weeks and weeks of trying to make the classroom into a cell, and listening to my classmates talk about their organelles every day, I pretty much understood, like,
All right.
yeah.
We get it.
Yeah.
Okay, somebody else? Any other?
I think it's very effective because we got to visualize all the organelles up close rather than just looking in the books.
And it was creative. We got to create our own own organelles, and that helped us understand better. So, yeah.
It's effective to other classes because when they come in, they get to understand and they get a visual of what they learned in the book, especially with physiology.
Good job.